in basic research focused on rational design and predictability of synthetic biological circuits. And we also do part of our research in applying what we learn from our basic research studies in order to apply what we learn to real world problems like bioenergy, environment, and so on. And we will talk about some of these a little bit later. So, as I said, I'm assistant professor there, and I'm also visiting scientist at Institut Pasteur, in which I do part of my activities focused on single cell analysis. So basically, it is an activity that is focused on the better characterization of some of my strains that I work on. I didn't say it, but I'm interested in bacterial synthetic biology. So all the things that we see in this presentation are focused on mainly on E. coli. So it is on bacteria, and it is also good to study single cell to make single cell analysis with the proper tools as I do here. So a part of the results that I'm showing are are taken from my team, and a part of that are on microfluidics analysis, as we'll see in a while. So I think that Paris doesn't need Google Maps to understand where it is, but Pavia normally has to, because nobody knows where it is. It is a small town near Milan, and so it is settled right there, just to let you know. It is a good place to stay. We have even a bridge like that one, and it is a picture in which there is sunshine, but normally we have a fog, and yeah, it's a nice place to stay in any case. Okay, so talking about some motivation. So why are we interested into rationally designed synthetic circuits? So why are we focusing on predictability? So basically, because cells are great, so they do a lot of things for us, and it is important for us as a biological engineers to understand how we can design biological circuits in order to rewire the sensors, the actuators, and the information processing parts of the cells in order to make customized functions. So that's the goal. The main issue is that we have a limited success right now in the composition of predictable biological circuits. And the reason is that cells are affected by a lot of unpredictability sources, as we'll see in a while. And yeah, so we need to find methodologies to improve this success. In the last few years, there have been a lot of teams proposing some automated platforms for construction, for characterization, as foundries for biological circuits in order to increase the throughput of the construction of biological parts. But they still rely on trial and error, because they increase the throughput to try a lot of candidates for the constructed circuits. And it is a problem, because platforms are not accessible to everybody, first of all. And then if we rely on trial and error, we still do not have the possibility to rationally design circuits with a high success. So designing a circuit means composing DNA sequences that encode the functions that we want, and then we have to insert them into the cells of interest. So that's the main task. So how can we do to better address the rational design issue? So we can learn from our failures, first of all, and it is important to that's going to be with us during the presentation. So it is important to learn from our failures and to study non-functional case studies. So it is good to fail, because we can learn a lot of things from the weird behavior of some of the biological systems that we construct. And eventually, we need tools to analyze them in order to see rationally what is the failure, and eventually to avoid that in a second time. So in our experience, we had a lot of failures in synthetic circuit design, but probably one of the most, one of the major causes is the cell stress that is caused by the heterologous gene expression that exerts a unnatural load for the cell. This is why we want to express something that is not natural in the cells. So we insert biological circuits and we have heterologous expression. So this kind of expression level that we engineer for different genes of our circuit could be too much for the cell. So that's the point. And in order to obtain a higher success rate in the circuit design, we must have the tools to measure it quantitatively, first of all, then to predict what happens by combining components that are burdensome, and eventually to mitigate its effects. So that's the whole story that we would like to do. So in this presentation, this is divided in 4 parts. First of all, I will show some examples of biological circuits that are affected by cell stress, and so they do not do what we want. So we will see 3 failures, I guess, in order to try to learn something from them. Then we will see some experimental measurements and also mathematical modeling tools that will be useful to characterize the stress in the circuits. And then we will move to how to mitigate cell stress by using some good designs of biological circuits. In the last part, we will focus on, quite briefly, actually, on some of the applications from our lab in which non-stressful functions are very good and provide some benefit to our to solve our issues. Okay, so first we study the effects on 3 case studies of cell load. So first, before showing them, just the main rationale of the construction and study of synthetic circuits. So our synthetic circuits are just combinations of transcriptional units in which we have genes that are regulated. We have promoters and ribosome binding sites that regulate the expression of the protein, and we deliver everything inside, in my case, bacterial cells. So all these genes are expected to interact with each other as we want, okay? But the problem is that if the cell is stressed by one of more components, the circuit will operate by interacting with the cellular context because it is not orthogonal, and we could deplete some of the cellular resources because of one or more of the genes in the circuit. So the main problem is that if we design a biological function that is a set of interactions that happen in the circuit, we have our interactions that we want, and we also have some unwanted interactions. So in the slides they are called hidden interactions, that are like repressors that occur when we don't want to occur, and so on. And this is mainly caused by the depletion of some of the resources that cause all these behaviors. We will see them in a moment. Okay, so just a definition that I will use in this presentation. So this is not universal definition. I define load different from toxicity because for me the load is caused by the expression of the genes that causes some burden for the cell. So it is caused by resource limitation and it is caused by expression. While toxicity is not caused by expression itself, but it is caused by the gene product that does something to the cells that we don't like. So just because we will see this distinction later on, and I wanted to make sure that it is clear. Yeah?
I understand this conceptually,
Mm-hmm.
but in real life, they are very much
Overlapping.
very
Yes.
much overlapping.
Yeah, exactly. We will see in a while by studying like a DEDCAS9 gene. that is expressed, and we will see the contribution of the expression itself, that when it is too high, we detect stress with the tools that I'm showing in a moment. And we also see some toxicity in order to, so in terms of phenotype in the cells that is not connected to expression, but it is connected to something already known in publications that is exerted by the DCAS9. And this is just a spoiler for later, but it is so it is not a universal definition, of course. They are, of course, overlapping because one causes the other, but toxicity could be more specific to the gene itself. While
I mean, my model is, if you have you saturate and folding capacity in the cell, right? So these are resources, right? But if you have a gene that depends on, like, an exogenous gene that depends on the system, and in its absence or when it's unbalanced, some metabolite changes, right? Then you get the indirect toxicity of the metabolite because you depleted the exogenous source.
Right?
So,
it's more indirect. So
of course. That's why I'm saying they are highly coupled. It's not easy to say it's.
I agree. Of course. So this is just the first example we will see. So the effects of load could be at different levels. So they could be mild to severe effects. The first example is quite intuitive and also quite simple. So I'll show it as the first one, and we will see some mild effects. So we have a simple gene expression cassette that is an inducible circuit that is a PILAX promoter. So it is an inducible system in which the input is the concentration of HSL, that is a homoserine lactone, quite broadly used in synthetic biology. And so this is the input, and as an output, we have a fluorescent readout with RFP. So we have a red fluorescence, and we expect that this function is an increasing function of our concentration of the input. So so we have just one module, and it is quite intuitive to have an increasing function of HSL because of the structure of the circuit. So what we see is that when we take the plasmid encoding for this function, we insert it into the cells, and we characterize in plate reader experiments like this. What we see is that we can measure the cell density and the fluorescence protein expression, and we can measure the steady state transfer function of this device by combining cell growth and protein expression to compute a steady state expression rate per cell. So these curves are the dose response curves in which we have on the x-axis the concentration of the input, and on the y-axis the output in terms of fluorescence. So what we see is that this circuit gives the black curve that is an increasing function. We are happy with that, but actually, when we re-engineer the cells and we insert a second plasmid, that is the one here below, I just call it load because I don't care what is there, because actually it is almost an empty plasmid. It is just an extra load for the cells to be replicated and probably expressing the antibiotic resistance in that. So when we insert an extra load that is not interacting with our circuit, what we see is that the output of the system decreases, and we have the red curve here. So qualitatively, it is still good because we still have an increasing function that is qualitatively as we expect. But quantitatively, we have a twofold decrease in expression. So as I said, it is quite intuitive because, of course, we add the load and we decrease the expression of something. Okay, this is just a first site of all these problems. And this decrease is not correlated with the copy number variations, because we have also characterized the copy number of these plasmids, and they are quite stable. So they are not decreasing because of the load. So the load is causing something.
The second option.
Yeah, really? Okay. I have never read about it. Okay, so I just click X. So yeah, okay, thank you. All right, so this one is the first example, and it is mild. So let's go to something more severe as an effect. So we have a more complex system in which we have a layered interconnection of different modules in order to form a transcriptional cascade of genes. So by knowing the transfer function of each of the modules, we can predict the final output, at least qualitatively. So in this design, we have at first one input module that is more or less the same as before, and it is an increasing function. And then we have two decreasing functions that are two NOT gates assembled in series. Okay, so with a yes, not here. Okay, let's try. With a yes, not combination here, we have a yes in the output because we expect an increasing function. We expect to have an increasing function of our input because the two NOT gates neutralize themselves. Okay, so we have constructed different designs for these cascades by using different components. For example, we have in this example the first module that is same as before, and we also have two NOT gates in cascade by using the Lac ion that are quite traditional regulators in synthetic biology that are used to repress the cognate promoter in order to implement a NOT gate. And we also have fluorescence as a readout at the end of the circuit. What we see is that this combination gives a good result, at least qualitatively, because we have an increasing function as we expected, but just, okay, so let's try the third one. Okay, so by using exactly the same components as before, but just switching the two NOT gates, we had a completely different result, and we see that the function is broken. So we see that the RFP is now a decreasing function of our input concentration. And it is absolutely unexpected because we have broken the logic, at least.
And this is independent of the ACAT and I.
Yeah, so I'm using here the regulators without the inducers of themselves. So the only inducer here is HSL of the first module. Okay. So I'm not tuning the two NOT gates. So I just use them as repressors. Okay. So in order to better understand what happens here, we will see that a debugging process for this circuit will reveal the guilty here, and it will reveal which is the most burdensome component of this circuit. And we will see in a moment. I just mentioned the third case study that is a little bit more complex, not because of the architecture, but because of something else I will see now. So we have a two-module cascade in which we have an input and a NOT gate downstream. So we expect to have a NOT gate overall. Okay. So we have implemented everything with a slightly different design than before. So we have an HSL inducible input, and then we have a NOT gate composed by a dCas9 and a single guide RNA that implement a NOT gate that repress the promoter upstream of the RFP. So we expect to have same behavior as before with the NOT gate, in which instead of that R and Lac I, we have the dCas9. Okay, just and we expect a decreasing function that is exactly what we have here. So we have implemented different of these circuits by changing the strength of this promoter. So this pconst is a member of constitutive promoter libraries that are assembled there, and we expect to have the dCas9 repressing them because we programmed the guide to do so. Okay, so we have constructed different variants of this one, and here we have the results for two curves given by a medium strength promoter and a very strong promoter in this case. So basically, and apparently, all of them seem to work well because we have a decreasing function. But the problem is a little bit more in depth because these curves are too steep. So we don't expect this kind of steepness because actually the dCas9 here targets the output promoter that in just one binding site. So we expect not to have a really steep behavior. We expect to have something more smooth, and it comes from the knowledge of the Hill equation, in which we can estimate this coefficient that is called the Hill coefficient in yellow, that is related to the steepness of the curve. Okay, so we expect that the steepness of the curves by changing the promoter is the same, and it's also quite low because we are using the dCas9 component. Okay, what we see instead is that the Hill coefficient is very high, so we have a high steepness, and we also see that this steepness increases with the strength of the output promoter. So this is quite unexpected in this case. Okay, so this is just the summary. So end of the first section, in which we just saw observations of weird behaviors. Okay, so let's see now, at least for second and third case studies that are a little bit less intuitive, how to deal with the behavior that we observed. So let's go to the experimental measurement and modeling of cell stress. So first of all, we would like to have some tool to experimentally measure the stress in terms of load caused by circuits on the cells. And in order to do that, we have applied a method quite recently proposed that is based on the expression in the circuit of a constitutive reporter gene. So the basics, the idea here is that if we have a constitutive cassette of a reporter gene, it is expected to be constant throughout the functioning of the circuit, throughout all the life of the cell, and so on. But if we assume that the limitation of resources caused by one or more components affects globally all the proteins of a circuit, of the host itself, so we can use this constitutive reporter as a proxy of cell load, because we expect to see a drop in its expression because of the limitation of resources. Okay, so we expect to see a constant GFP, for example, when we have no load, and we expect to see a drop that in the GFP when we have a load, and the entity of the drop is related to the burden given by one or more components. Okay, so that's the rationale of this, and it is quite convenient because we can do traditional measurements of fluorescence. So it's good to have that. Okay, in order to close the loop, we need also some tool that is based on predictions. So we have there have been a lot of models in the last few years proposed to understand, to study cell load, but here we have used one of them that is very simple, and very conveniently it is also identifiable from experimental data, and it is very important when we want to debug everything. So it is not just a simulation tool, it is it can be parameterized by experimental data, and then we can predict what happens in our case, not in a general case, to simulate what happens by changing parameters and so on. Okay, so the model is based on Hill equations, as we saw before. Hill equations are traditionally used to describe activation and repression functions in circuits, and the equation above is just the model without load, the traditional one. In order to add to describe the load in a circuit, we can still use Hill equations and add a denominator that is basically a penalty for all the Hill functions. So just to make sure, so the Hill functions describe the expression rate of a protein, for example, and in the no-load model, it is something like that. In the model with cell load, we have the Hill equation that describes the maximum expression rate of a protein, and this maximum rate is penalized by a denominator. So we virtually never reach a maximum, because this maximum is affected by the expression of all the other proteins of the circuit. So the denominator is based on a weighted sum of the expression of all the other genes in the circuits. And the weight of this sum is called the resource usage of the given gene. So we have a single parameter that could be fitted, that could be estimated from fitting the data, and this parameter is related to the load caused by each component. Okay, so this is quite important because we can debug.
I can understand mathematically why it's easier, but why logically?
Because it is an assumption, of course. Yeah, it is an assumption because. So
I mean, given the nonlinearity of all your system, including the Hill function of every you know of, right? I mean, it's highly unlike. I mean, mathematically, it's the only way probably to work with. Right? But
yeah. Sort of. So basically, this kind of model has a mechanistic explanation that is not. It's not from us, but it is from the original work, of course. The very strong assumption is that the denominator is the same for all the proteins. So that's the real. It could make sense because theoretically, because every protein feels the same burden actually, but sometimes it is not. Of course,
it makes sense if it's only the sigma of all expression proteins, independent of their character, their pathway, their folding rates, whatever, right, are the same. Right?
Yes,
So
yes.
Copies or 500 copies, you have a 0.5 effect.
Yeah. So of course, yeah. I see.
But everything that you showed us until now showed us that this is not the case, right? So
Yeah. So here, the weight is always multiplied by the expression rate, of course. So it is not independent of the copy number, of the transcription rate and translation rate. So it is dependent on that.
The absolute copy number of proteins. It's a sigma of.
Yes. Okay. So we can use both tools, so mathematical tools and the experimental measurement to fit all the collections of our circuits, basically. Here there is just an example to see what happens, but all the same procedure has been done with the other components of the circuit in combination or alone in order to identify the models and understand which are the burdensome components. So here there is an example of one of the circuits in which we have an input and we have a NOT gate downstream, and this NOT gate is based on that R. So what we see from the data, at least, is that the RFP goes down as we expect, and this is mainly because of the Hill function that fits this data well, and they are decreasing. The GFP output is something more complex, in which we have at first, for high RFP values, we have a GFP value. GFP, I just recall that it is the proxy of cell load. So we just see GFP constant if we have no load. So here we already know that there is some loading component. Okay, and in the first phase we have a little bit drop of GFP compared to its maximum level because of the high RFP expression that causes a little bit of load to the cell. Then we have the maximum of GFP, and then we have a larger drop. So this larger drop here is caused by, so it is in correspondence of the HSL inputs in which we have high TETR expression. Okay, so we can understand from this data, at least the experimental data, that when we overexpress TETR, we have a load. Okay, already from the data. Then by using the model is able to take into account all the proteins expressed in the circuits and to parameterize these equations that we see here. So from the fitting process of all this kind of data, we can do like a ranking of all the genes considering their resource usage. For example, we see here the 3 proteins of interest in the circuit, and we see that the guilty was TETR, based on these parameter values that have been estimated. And as we see here, so what happens is that TETR alone is, so not alone, but virtually any protein in the circuit could make a load for the cell, but TETR this time does much more than the others. So what happens is that TETR has many hidden interactions in which it is not just the repressor of its downstream gene, but it also represses a lot of other genes in the circuits. Okay, and so with the model resulting from this process, we could predict quite well that the function is not increasing anymore. So the red curve is just a prediction, so it is not a fitting here, and it is quite good to see that we are able to capture the effect. So we are able to measure the load of the components, and we are also able to capture what happens when we combine burdensome components to each other. Okay. In the other example, I go quite briefly with that. The guilty was RFP, and it was because its expression was very, very high. And what we saw was that RFP, by being the burdensome component, was the repressor of its repressors. Okay, and what happens is that we have a sort of feedback, and this kind of feedback makes a very steep switch when we have a high expression rate for RFP. Okay, so just by increasing the RFP expression level, we have steeper switches because our components are all affected, and the switch increases the Hill coefficient. And this figure is just a simulation showing that the Hill coefficient increases as a function of the J parameter of RFP, but also with the expression rate of RFP. It depends on what we are describing. Okay, so in general, it is cool that we have methods to quantify everything of this, and it is cool that these methods are also have somewhat predictive power to understand qualitatively and also quantitatively what happens when we combine some components that are problematic. Okay, a quite intuitive question is, so why do we have to use GFP? So we could use, at least intuitively, growth rate. So growth rate is a traditional index of cell load, because the lower the growth rate, the higher the load. Okay, so quite intuitive. The reason why we're not using the growth rate with that is that GFP is much more sensitive to understand what happens inside the cell. And this is, on the left, we have one example demonstrating this. So we have a simple input module in which we produce RFP, that is an increasing function of our HSL, that is the input as usual. So we have a RFP that is increasing as expected, and we have GFP that is slightly decreasing because RFP is causing somewhat a load for the cell. However, the growth rate here is basically unaffected. So we can see that GFP is able to capture something that growth rate is not able to do. Okay, and we did the same for basically all the circuits that we had in collection, and we had the graph on the right that is very low. Yeah. No, so it is plate reader experiments like this one. So we just cultivate the cells inside the multi-well plate, and we insert in the plate reader that acts also as an incubator, and we measure over time for every like 5 minutes absorbance, fluorescence.
You take the maximum slope.
So we just define boundaries for the absorbance values that are already characterized in our system to be more or less linear. Okay, and then we do the logarithm and we do the linear regression for that. Okay, so it is not the maximum, because the maximum was, yeah, so we tried with that, but it was not very much informative, so it was not stable compared to, like, for example, flask experiments. If we do flask experiments, it is the best option, but yeah.
And if you try to correct carrying capacity, so maximal OD, what do you get?
Okay, so
because that's a bit more.
Yeah. So the maximum mass that the cells could do. Yeah.
How many cells do you manage to do per kilo of sugar?
Yeah. So it's interesting. So I've never done it, also because we normally do everything in the exponential phase in order to reach a steady state gene expression, because then later on it is not stable anymore in terms of GFP, RFP, and so on. So, but
if you have your curve, even if you don't reach completely, right? Just by fitting a logistic curve. Estimate the final OD.
I can think about that.
In our experiments, this fits better.
Yeah. It's a good idea. I just have to make sure we're not out of the linear range of the plate reader, of course, because So it is not the best option, of course, to do multi-well experiments, but they are really high throughput to do everything. So it's the way we do normally with this service. But yeah, it is quite interesting to check it if we have the maximum one. Yeah. Okay. So on the right, we just have a motivating curve in which we see that the correlation between GFP and growth rate is very low in all the circuits. And what we see is that also the range of variation of GFP is much wider than the range of growth rate, meaning that it is probably the most sensitive index that we could compute in this case. Okay, so in order to have a little bit deeper understanding of what happens, so as we said in the last answer, we have done everything in plate reader by now, but we are also interested in what happens at the single cell level in order to estimate the per cell growth rate, fluorescence in red and in green, and to see what happens to the single cells. Okay. So to do this, we are using microfluidics and live cell imaging, and these are the activities I'm doing this in Pasteur. And to do this setup, I basically use a microfluidic chip that is similar to a mother machine device. For those who are not familiar with the mother machine, this is the mother machine. So it is a microfluidic chip in which we have very tiny channels. These channels are more or less the same size as the rod-shaped bacterial cells. So all these channels have same height as the dimension of a cell and same width as the dimension of a cell. So these channels could host the growth of cells in one dimension. So in each channel, we could have like 5 to 10 cells, depending on the size and on the length of the channel. And in all of this, so the cells could grow in a monolayer. So it is good to not to crowd them and to keep them in focus. Okay. So these microchannels are just this centipede that we see here. We are using a little bit different setup compared with the traditional mother machine that is very widespread in microfluidics. So we are using a dual input mother machine in which we have 2 input ports instead of 1. And these 2 input ports are, where is it? So the 2 input ports are connected with the reservoirs of our media of interest. So for example, one is the media alone, and one is the media with the chemical inducers. And in order to tune the concentration of the chemical inducer that goes to the cells, we can use a junction that mixes the two fluids, okay? And we can also use pressure controllers in order to mix them predictably, programmed via software, for example. So the junction I'm talking about is this dial-a-wave junction that we see here, that is a little bit more complex than the traditional T-junction, because it has two inputs with the two input reservoirs and three outputs, in which the two lateral ones are just overflows, and the central one is the one in which we want to tune the mixture proportion. Okay. So, for example, if you see the animation, we can tune the input pressure to define a good proportion of the input media in the central channel. The central channel then goes to the cells, and they should have this stimuli. Okay, in order to, so since with microfluidics we are in laminar flow, the two fluids are not mixing immediately, so we need a long enough path in order to mix them by only with diffusion. So we need a serpentine. That is the reason why there is a serpentine like here, in order to have the good path to mix the two fluids and then feed the cells with the media that is mixed. Okay, so this chip has been, so we have constructed the chip and we have set up experiments. So this is basically the experimental setup that we carried out with the help of the biomaterials and microfluidics core facility in Pasteur. So we have a thermostatic chamber in which we can put our chip, and we have pressure controllers right here, are the blue modules that are connected with the reservoirs that are basically the two falcon tubes above that are pressurized, and they are connected with the chip itself, as we see in the corner right here. Okay, and this is the experimental setup for this mother machine. A quite important part of the work is also to analyze the data and not only checking the movie. No?
Yeah, no, we developed the initial mother machine was made in our lab. We have all the software available to do this freely available, and it's always funny to see how people reinvent the wheel again and again and again and again.
Yeah, so we haven't invented anything basically. So we have tried with our softwares, but many of the softwares were specific for phase contrast imaging, and in our lab we had the bright field. So we not only the bright field, but I was using the bright field, and all the softwares couldn't work.
Now you tried ours, but we are 5 meters away, right? Okay.
No, but it's very interesting for me to have something already done.
Put you in contact with you.
Yeah, would be great. Thank you. So basically, we have used a published method that is based on artificial intelligence and deep learning in order to carry out segmentation and tracking. And this is important because we could track all the cells growing in the channels and to quantify the per cell fluorescence and also the division time in the channels. Okay, so this is mainly the setup, and so as an overview of the experimental data that we have, so this is just not the analysis result, but it's just the experimental data. We have so I just show here the results for the circuit on the left, in which we have an input and a NOT gate. And this NOT gate is burdensome. Okay, so we have made experiments in which we have at first no inducer, and then we have perfused an inducer at a saturating condition, and then wash out for still 6 hours then. Okay, and what we see here is more or less what we expected. So at first we have red and green expressed, so no particular load and RFP expressed. Then when the inducer is added, we repress RFP as expected, and we also see that GFP is coming down. So we also have a drop in the GFP as we expected for the load that is caused. When we remove the HSL from the culture, we see that, so here qualitatively at least, we see that GFP is turning on again, meaning that the cell load here is reversible. So we see cells that are with better health state than before because we turned off the expression of that R. So here we can see that GFP is recovered, but RFP is not recovered yet, meaning that we still have that R in the cells that is not washed out due to cell division. Okay, and it is quite important because at least we can decouple cell load with toxicity effects.
What's your division time?
Next slide. More or less, so doubling time is the time between two splits of the cell.
So your suggestion is that because within two divisions you expect to get R. So you're so much overexpressing it.
Yeah. It's what I also saw in plate reader experiments in which we have to subculture the recombinant strain because otherwise we don't see anything turning on again. So if we start with the theta expressed like this, it takes a much long time to wash it out. That's, yeah.
Suggest that if you just put very bad RBS in front of it.
Exactly. That's the next slide, but I don't want to run. Yeah, of course. So this is more or less what we expected, and we have decoupled. So we have identified that the problem was that R, but it was not the product of that R, the protein, but it was its expression. Because exactly when we turned on, we turned it off, we have a recover of the health state of the cell. So that's the idea. Okay. By looking at some quantitatively per cell analysis, we see more or less the same. So we have RFP that is decreasing towards 0, except for some oscillations at the beginning. And we also have GFP that when we apply the input, it is dropping, as we see here, not to 0, but it is slowing down its expression. And then when we remove the input, we have a recover, okay, as we expected then. Okay, so just to confirm what we saw before, the division time was not affected too much because in the 3 phases, induction, no induction, and so on, we could see more or less the same growth rate, okay? So this is the time between 2 divisions, and it is more or less constant with these phases. So it is a preliminary analysis because we just take into account a few channels and so on, but then we will see something in broader scale later on. So not in this presentation, of course. And on the left, we have more or less the same information by checking the mother cell, that is the cell at the very top of the dead-end channel. It is called mother because its daughters stay there for all the experiment. Okay, and what we saw is that there is no particular sign of slowing down the division. Okay, so this picture is just the area in pixel of the mother cell that increases because the mother is growing, and then it decreases abruptly because of the split. Okay, so in general, it is good to have all these methods that. Of course, we have seen some results that confirmed what we had before. And it is also good instead to avoid this kind of effects and not always to measure the stress and so on, but also to mitigate what is happening. So a quite intuitive option here is that, okay, I have a burdensome component. Let's weaken this expression. So as he said before, we can, of course, decrease the efficiency of the RBS. Yeah. It's what I'm doing now. Exactly. But not yet with just one channel, but I think that it is just statistically not significant. Okay. Yeah, exactly. It's something, yeah. So well aligned. Yeah. But of course, by checking a couple of channels, it's probably not enough to see. Yeah. I have a lot of dead channels because, but because efficiency is not 100%, not yet, but because of the operator that is me. Then yeah. So an intuitive option here is to weaken the RBS to decrease the expression of a loading component, and we did this. So we weakened the RBS of that R, and we actually recovered the good function that we wanted. So instead of having this the decreasing function that we saw before, here we have a slightly increasing function that is at least more or less what we wanted. Okay. The problem with this approach is that we also change the transfer function of that R, because if we weaken the RBS, we have a larger switch point, a different expression level in the output, and so on. So if we want to have a circuit that behaves as we want, we cannot use this approach because it changes the behavior. Okay. So in order to try to fix the problem, we have used in our lab the Crispr interference modules to replace burdensome components. So the basics of this approach is that if the main cause of cell load is translation, we can use components that are not translated. For example, guide RNAs that are just transcribed, and so they're not expected to exert a very big load. Of course, in this system, the DCAS9 expression doesn't have to be a big load for the cells, because otherwise we are not solving our problem. So we tried to fix the cascade that we saw before with this kind of components, and first of all, we had a lot of work by tuning the expression of the DCAS9 to non-toxic levels. So here we have the first design of our inducible system for the DCAS9 that had basically all the signs of load and toxicity. So we had it assembled downstream of an HSL inducible promoter, and we had a decrease of GFP and also growth rate as a function of the input. So not good at all, and we could also detect some toxicity due to some filamentous phenotype watched at the microscope for the cells. That is a sign of toxicity of the DCAS9 at too much high levels. And it is basically happening because of some off-targeting without any guide in the genome and so on during the scanning of PAM sites. Yeah. No, okay, so the characterization right here in this slide, there is no guide RNA. No, actually, no, it is not necessary. It is not necessary. So the final system is expected to have some constitutively expressed DCAS9 that is enough for the NOT gate to operate in on and off levels. So the sgRNA should be off when it is not expressed, and when it is expressed, there should be enough DCAS9 to bind it and repress the target promoter. So I don't know if I answered the...
I'm just wondering if DCAS9 is hitting in some cases, therefore it's also...
Yeah.
And so it's
of course, in a few slides that there is something, and then you tell me if, yeah, okay. So this is the first, design. So after optimization of promoter and RBS, we could remove all the signs of cell load in the DCAS9 expression cassette. So we have healthy cells with a normal size, as we see here, and we have a load that is almost null here, with a constant GFP and a constant growth rate, and we have a good repression of different targets that we tried with the RFP this time. So in this case, we could try to fix our cascade by replacing the TET-R with the sgRNA. And of course, as usual, the first design didn't work because the first design is this kind of curve that we see here that is flat completely. So actually, our single guide RNA was too much efficient and basically clogged the promoter at all the conditions. Okay, so the approach here was not fully rational, so I'm not happy with that, but it was effective to weaken the guide RNA by introducing some point mutations in the guide. So at first the guide is perfectly matching, and here we engineered some degenerate sgRNAs to weaken the guide. And then we screened a lot of components and we selected the one that had a good repression range here. So the final candidate had a transfer function that is not perfectly the same as the one that we saw before. So the dark curve is the transfer function of our CRISPR system, just the NOT gate, and the one that we wanted to mimic is the dotted one. So not exactly the same, but more or less the same range. And we, yeah.
The dynamic range.
Mm-hmm.
Right? So the dynamic range is formed lower.
Yes,
actually.
it's true.
I mean, it looks on the graph, it looks very close.
Yeah,
right?
yeah.
But if you look at
it's not
5 divided by 0.05 compared to
yeah, exactly. So there, it's true. So in general, as an approach, so here it is also true that we screened not thousands of components, but tens. So it is also probably so not a perfectly matching one, but we were also interested into what should happen there. So when the p-TAT is off, we were not very much worried about that because the LAC-I doesn't have a much high sensitivity for the output promoter. So even if we have this, where is it? This difference does not make much sense for the input of LAC-I. So I don't know if I was clear. So not leakiness. So leakiness here, true, but our LAC-I was the opposite. So it needed a lot.
The idea that the LAC-I component.
Yes, in this case, yes. That's why we were happy with that, but not happy with the higher state. Yeah.
As long as the LAC-I does not use the higher states.
Yes, of course. Yeah. So it was a little bit application specific in this case, but in general, as an approach, if I had to do something more general, I would have selected many more point mutations and screened a lot of components there. But in this case, pragmatically speaking, we were happy with this solution, and it could restore the expected function that was an increasing function. And this was as expected, and it didn't have any particular sign of cell load with the constant growth rate and GFP throughout the all induction levels. And last but not least, we were very happy that it was also predictable by means of a mathematical model that is a traditional model. So without using any cell load modeling with the denominator and so on. So traditional modeling with the the components characterized separately could give more or less the same output as we expected. That is quite cool. Okay. Just to let you know, of course, related to the previous question, we have not solved all our problems by using the CRISPR components. Probably they were solved in our system because we had just one guide. But in general, if we want to use it as an approach, it is not very good to have many guides that have not ribosomes to share because they are not translated. But this time there is a shared resource that is the dCas9. Okay, and if we consider this, we still have a resource limiting problem. I'm not going deeply into this, but we have addressed this, of course, in collaboration with a group at the MIT, by proposing a control theory-based approach in order to stabilize the dCas9 expression level based on the demand of the guides that are expressed in the circuit. So the meaning of this slide is not going very deeply, but it is that not all the problems are solved with the CRISPR. We still have the resource limitations, but we have tools to limit, to mitigate also this level of limitation. Okay, so this is the point. I just finished with very briefly with if I have time, so with the last section in which I just show 3 different examples from my lab of more practical problems. So we're not here talking about GFP, RFP, and so on, but something more applied to some areas like energy, environment, and therapies. So we see something that have in common that we could have a low load in the circuits, and this low load gives us the possibility to engineer predictable functions and also long-term operating functions, because so the predictability was not the only problem with the load. The other problem is that the function becomes unstable if the cells are stressed, of course. Okay, so in the first example, I just show an engineered strain of E. coli that has been modified to overexpress an ethanol production pathway, and we are using it to ferment dairy waste materials that are rich of lactose. And of course, the first versions of the strains were poorly functional because of the stability. So they had the ethanologenic enzymes expressed too much, and this level was not compatible with the long-term cultivations and so on. So in our modified strain now, we have an ethanol production pathway that is given by 2 heterologous genes, and also given from the knockout of some competing pathways in the fermentation metabolism. And the good thing here is that this strain could support the growth on the very long term. So we cultivated it basically for 1 month without any selective pressure, and we could have more or less 100% of the cells still showing ethanol production phenotype during all this period. So this is a good example of a stable strain that has been engineered being gentle, and as a response, it can be reliable for all this time. Okay, so in this data, the ethanol production pathway is enoplasmid. The plasmid is not subjected to selective pressure, and it is good that all the colonies then are maintaining the ethanol production pathway. The colonies here are just plated on a McConkey plate, in which the logic is, if the ethanol production is functional, then we have low amount of acids produced by the strain. Otherwise, the strain gives like purple colonies that are an indication of high acid metabolism as an output. Okay, so this is more or less the logic. And by having this stable strain, it is very good that we could move to something bigger. So it's a quite good story, because this project actually started with an iGEM project many years ago, and it is very good to see it was scaled up, and now we have a good reactor working on 200 liters, fermenting dairy waste and producing ethanol, and I think it is a good showcase for iGEM projects from, yeah.
So what did your plasmid looks like? What's like the promoters?
Yeah, so it is a low copy plasmid, so 5 copies, PSC 101 origin, and I have the 2 recombinant genes, codon optimized, under the control of very strong RBS, and the promoter is just the PILAX, traditional PILAX, with no induction.
And what does it make in non-stress?
The choice of the promoter itself, because other candidates that we had actually had a higher production rate of the genes.
Yeah.
So basically, in this project, we wanted to overcome some limitations in the literature, in which the people saw some very high demand of transcription for the 2 genes when they were not optimized for translation. Okay, so the point here is that this plasmid had a low toxicity and burden for the cell, while other candidates that had different copy numbers or different promoters could have a higher load, basically.
One of the limitations in the pathway is that the
Yeah.
So this has to do with the tuning of the ratio between PDC and ADHD.
So, okay. Yeah. So we looked at it, but actually we so it was not a toxicity by acetaldehyde this time, because the tuning was done basically in traditional LB medium that doesn't have any fermentable source, and so it couldn't happen. Okay, because acetaldehyde needs a lot of pyruvate in this at the end of glycolysis to be expressed in this time. And so what we concluded from those experiments or that by checking all of this with the traditional LB medium, we are not checking the fermentative pathway. We are just checking the expression of the proteins. Okay. So, yeah. Now the final strain is not the one with the plasmid. We have done a lot of work again to integrate all of this in the chromosome without any pressure, antibiotic resistances, and so on, in order to be compatible with industrial use, actually. So that's but the point is that we had to engineer the expression rate of these proteins to be gentle, and in response, it could last for a lot of time. Okay. As a second example, this time we had another circuit that needs to work continuously for a lot of time. And in this case, we are in another field, and we are happy to construct engineered living materials by means of bioprinting techniques that can make us obtaining some materials with included some bacteria that have been engineered in order to show some biosensing capabilities, for example. Okay. So we have a bioprinting pipeline in our lab that produces, here we have like grids, but we can produce different things. And the field we are working on is biosensors, in which we would like to have long-term functions, meaning that a sensor should be placed in some land or whatever, and it should be working for a given time, not like one day, but much more. And the cells should be valuable, and the function should be stable. The first options, the first designs of these sensors were in, for example, high copy plasmids, or they contained the TETR, or contained a lot of other things that didn't make the cells reliably working. But with designs like this is just a spot. I'm not going very deeply into that. With the optimized design of what happens. So by choosing low load components, we could ensure long-term availability for a couple of weeks. And for all these couple of weeks, we could analyze the colonies that could be extracted from the material dissolved by sodium citrate in this example, and they could be counted to check availability and the function itself by checking the color if the biosensor is working. So also this time, more or less the same message as before. We can be gentle this time, and as a response, we have a long-term functioning of our engineered system. This is the very last example in which in this activity we are engineering probiotic strains, basically, in order to act as Trojan devices for different pathogens, actually. So in this kind of system, we are engineering the CRISPR to target different antibiotic resistance genes by silencing them. So the basics of this idea is that we have a donor strain that gives to the pathogen receiving strain a plasmid in which we have the CRISPR components, and these CRISPR components target transcriptionally the antibiotic resistance of the pathogen. So in this case, in order to have this strategy as a silent strategy, not using the cutting of the genome with the traditional CRISPR system, we are using CRISPR in order to make it silent, I guess. So we insert the plasmid, we repress the target antibiotic resistances, and then we rely on traditional antibiotic therapy to kill the pathogen, basically. That's the main idea. And in this case, we were successful by using the low burden Dcas9 cassette that we saw before, because otherwise, if we have a high burden cassette, it is not meant to be a silent strategy anymore. Okay, that's the main logic of that. Okay, so I just go to the conclusions. So in general, that are just a summary of what we saw. So in general, yeah, cell stress can break functions, and so it is not good to have it. And it is good, but it is good to have the tools to measure this and predict what happens with the burdensome components. Also with some single cell measurement platforms in order to check what happens at the single cell level. And it is possible also to design some low burden components that are mainly rational design, but not only rational design, which I'm not very happy with, but it's what we did until now. And of course, as we said, said in the last few slides, the applications can greatly benefit from circuits that are gentle with the host, because we can have more predictable and also more stable functions. Okay, I just thank, of course, you for your attention, and all the people in this slide, because they have contributed at different levels. So there are a lot of people at the University of Pavia in my group, and different people at the Institut Pasteur that helped the microfluidic design and the development of these activities, and the sponsors on the right that I would like to thank also. All right, so I think I'm out of time.
What's the effect of like simple perturbations like in media composition of the circuits?
In which part?
Like generally, like is media has a strong effect.
Okay. So I tried some, for example, in my burdensome model that is the Tatar, basically, I tried to cultivate it in different media, for example, in order with the assumption that okay, if I increase the nutrients, maybe it has more nutrients and it is not resource limiting, but I still see the behavior of the cell load that is going down. That is, sorry, not going down, but the GFP is dropping, and
So the trend is conserved.
Yes, so I don't have hundreds of examples. So I just have a pilot experiment in the microfluidic device, and the trend was exactly the same. Yes.
Like nitrogen limiting media, this type of thing that have not.
So in the experiments here, what I showed is more or less all based on M9 supplemented media. So it is the minimal one plus casamino acids and thiamine and glycerol and so on. And the rich one was LB. So with these two, so I haven't done supplementations and so on. So I don't know. What I feel is that there is an issue also with the more rich sources. Mm-hmm. But it would be interesting to study if there is some kind of like chemical limitation that we can
like the
complement.
Metabolism of the host, if metabolism changes, because like like a lot of like biases, like in real life conditions, like outside of the lab, like. Sure.
Yeah. So I don't know, because also in other complex environments, I have only worked on these two applications, actually. So here I have more or less the same things in whey permeate, so dairy waste, in which I see more or less the same as in LB. So there is some dependence on the media, but not overall on the toxicity. So if something is toxic in LB, it is also in M9 and in whey permeated. Yeah. Mm-hmm. It is not a general rule, of course. It's experience based on these examples. Mm-hmm.
Is there no other question? Actually, we're going to go grab lunch and then eating the other thing. So I'll also keep chatting there. Thank you very much.
Thank you.